thinking about orthosteric binding sites. So, for example, epidermal growth factor binds to the epidermal growth factor receptor at the orthosteric binding site, but it's possible that you would be able to come up with another agonist that would bind to the receptor at a different site, the allosteric site, and activate it as well. So today we'll talk about basically what allosteric modulators are and a couple of important models explaining their behavior. So the word allosteric comes from the Greek stereos meaning space, allo meaning other. So for example, like an allograft. So an allosteric modulator can influence the agonist or antagonist effects on a molecule as well. And probably the simplest way to think about the way that allosteric modulators work is that almost all receptors have multiple conformations. So oftentimes we think of a receptor as being an active or inactive conformation, and that what the agonist does is it favors the active conformation. So, but it's also possible that another binding site can modulate that same conformational change. So there may be multiple binding sites that can drive the change from the active to the inactive conformation. So again, the idea is that the allosteric binding site is separate from the agonist binding site, but that's not to say that they can't influence each other. And again, as we pointed out, this has to do with primarily with conformational changes in the target proteins. All right, so one of the most important models for allosterism was developed by Monod, Wyman, and Changeau. And this was the first model to be developed, largely based on oligomeric ion channels. The second model was developed by Koshland, Nemethi, and Filmer, and this is called the induced fit or lock and key model. And so we'll go through some of the basic differences between these two models. And I should point out that the reason there are two models is because that proteins have different behaviors, and so some proteins follow one model better than the other model. And of course, there's everything in between. So that's why there are two models, is because some proteins fit one model better than the other model. Now, what's common to both models is that they both assume that there's a conformational change in the protein. So the protein can exist in either the tense or the relaxed state, T or R. The difference between the models has to do with whether you're allowed to have a mixture of states within an oligomer or not. And also there's a difference in terms of what's thought to be important about the interactions between the subunits in the multimeric protein. And again, so these models deal with homo-multimeric proteins. So for example, dimers, trimers, or tetramers, or higher order oligomers of the same protein monomer. So concerted model, again, this is the Monod, Wyman, Changeau model. Key features are there's a conformational change at one subunit from R to T. But the important feature here is that in the Monod, Wyman, Changeau model is concerted. Concerted means that all of the subunits have to be in the same state, either the T or R. So in other words, if you have a tetrameric molecule, all four subunits have to be either R or T. And so when a drug molecule binds to one of the monomers, it can't just convert that monomer from T to R. It has to either have no effect or convert all four of the monomers. Now that being said, there is an equilibrium between the T and R forms. And so what the allosteric modulator does is it shifts that equilibrium. But again, in the concerted model, all the subunits have to be in the same state. Now, with respect to pharmacology, R and T are sort of abstract. So what we're usually thinking about, if it's, for example, an ion channel, we're talking about the ion channel being opened or closed. If it's an enzyme, we may be talking about whether it's an active or an inactive conformation. So these really have to do with regulation of protein function of different receptors. Now, the sequential model, or the Koshland model, also known as the induced fit, there is a change in conformation when the ligand binds to a protein. And so, and they can be just in one subunit. So that's the key difference with the concerted model, is that in this case, not all of the subunits have to change conformation, usually just the subunit that binds to the ligand. So again, it can change other adjacent subunits, but it doesn't have to. So what it does is the change in the conformation of, let's say, T to R in one subunit actually changes the interactions between the subunits. And those changes in those interactions can also favor conformational change in adjacent subunits. So in other words, it may increase the likelihood that they'll go from the T to the R state, but it doesn't require it. So you can have any possible combination of T or R subunits in the Koshland model, but there are changes in probabilities of which distribution of T or R subunits you'll have. So here are some figures that to give you an idea of what you would see with these different models. And so we're looking at up here, these little squares are the allosteric ligand. And so you start off in the Monod, Wyman, Changeau model, and you have no binding, but as you increase the concentration of the allosteric modulator, you can see that that now you're favoring the T, let's say square is T, the T conformation, and that all of the subunits have to be in the same conformation. So here you have all four subunits T, all four subunits T, and you can see that the ones that are in the R conformation, which in this case aren't binding any drug, all four subunits are in the same conformation. So as you titrate in more and more of the allosteric modulator, you can see that a larger fraction of your oligomers are going into the T conformation. So basically you're ending up with almost all squares. But again, you see an example here where this particular subunit is still in the R conformation, even though it's bound to an allosteric modulator. So again, the important point here is that all of the complexes are either to T or R. So even when there's an allosteric ligand bound, there's still some probability that the subunit will be, and in this case, in the R conformation. And again, T and R, in this case, are somewhat allosteric. The important point is that there are two different conformations, and that the allosteric modulator alters the probability that the subunit will be in one or the other conformation. Now below is the Koshland model, or the induced fit or lock and key model. And you see what happens is, in this case, you can see at the very beginning that some of these oligomers have a mixture of different types of subunits. So down here we see 3 of the R, for example, subunit and 1 of the T subunits within the same oligomer. As you titrate in the allosteric ligand, you can see that as it binds, it does favor the conversion of the subunit that it binds to, let's say, the T conformation in this case. So as you titrate in more, you get more and more monomers that are in the T conformation. But again, it's not required that all of the subunits are in that conformation. So as you titrate in more and more, you do get more and more in the T conformation. But again, not all. So in this case, you have, of this tetramer, you have 2 subunits with bound allosteric modulator, but you still have 1 subunit in this tetramer that's in the R conformation. So this is another, below that is another example. And this might be, for example, an ion channel that's in the open or closed conformation. And you can go through that and see what it looks like for either the Koshland model, the induced fit model, where not all the subunits have to be in the same conformation, or the Monod-Weyman-Chanchot model, where they do. And again, you see there are equilibria between these different conformations. But the important point is that with the Koshland model, you have the potential, at least, to have a mixture of different conformation types within an oligomer, but that's not true in the Monod-Weyman-Chanchot model. Now, the other thing to think about is that you can have different types of allosteric modulators, in the sense that there may be some allosteric modulators that will stabilize the T conformation. There might be other allosteric modulators stabilize the R conformation. So depending on which of those conformations is enzymatically active, for example, you might have an allosteric modulator that binds to a conformation that stabilizes the active state of the enzyme, or you might have another allosteric modulator that stabilizes the inactive state. So an activating allosteric modulator will be called a positive allosteric modulator, whereas the reverse will be a negative allosteric modulator. Now, you can see that in some cases, when the allosteric modulator is bound, that it stabilizes, for example, not only the round or R conformation itself, but it also alters the affinity of the surrounding subunits for the allosteric modulator. So in some cases, it can increase the affinity of the surrounding subunits for the allosteric modulator. That would be a positive allosteric modulator. Or in other cases, it can actually decrease the affinity of the surrounding subunits. So this is an example of cooperativity. So if you think about hemoglobin, as it binds, so it's got 4 oxygen binding sites. As it binds oxygen, it actually increases the affinity for oxygen, or the binding avidity of oxygen at each step. So it shows positive cooperativity. Now, the opposite of that would be a case where, let's say, you have some molecule that when it binds a ligand, the affinity for the second ligand goes down. That would be an example of negative cooperativity. So these models can explain cooperativity that's observed in these multimeric proteins. Of course, hemoglobin is a little more complicated because it's a tetramer, but it's not a homotetramer. So it's got 2 different kinds of subunits, alpha and beta. All right, so we're going to talk about allosteric modulators. We have to think a little bit about who cares. So why, if we can make orthosteric drugs, why would we want to learn about allosteric drugs, and how are they different? And in some cases, perhaps maybe better than orthosteric drugs. And so when I write exam questions for students, I oftentimes want them to tell me what the differences are, and why in some cases an allosteric modulator might be beneficial. Now, you have to keep in mind, though, that there are lots and lots of different types of allosteric modulation, and there are allosteric modulators that can do just about any possible thing that you can imagine to the receptor, and we'll see examples of that. So, so it's often a case-by-case basis. It's hard to make a lot of generalizations when it comes to allosteric modulators. All right, so obviously one of the potential advantages of an allosteric modulator is it doesn't have to compete with the orthosteric ligand, because by definition it binds to a different site. So what does that mean? Well, let's say that you have a molecule, let's say it's an estrogen receptor antagonist. And so what that means is it binds to the estrogen binding site of the estrogen receptor. And what that means is that in vivo, there's actually circulating estrogen, and so the antagonist has to outcompete the circulating estrogen. And so if you have a high enough concentration of estrogen, or if your antagonist doesn't have high enough affinity, you're going to actually have some estrogen still binding and activating its receptor. But if you have an allosteric ligand, that may not be true. So in other words, you may be able to have a ligand that binds to a different site, not the estrogen binding site, but an allosteric site. And in fact, that binding may block the function of the receptor, but not have to compete out estrogen. So that's one example. Another is that it can be more drug-like than orthosteric ligand. So let's say, for example, that we're interested in activating the insulin receptor. And of course, diabetics need insulin to live, so it would be actually very efficient and useful if we had molecules other than insulin that could activate the insulin receptor. So lots of receptors have proteins, or peptides at least, for their orthosteric ligands, and the problem with those is, in general, proteins aren't very stable, and they're not very easy to give as drugs. And the reason for that is that you can't give them orally in general. They have to be given by injection, and in general, peptides are just not all that stable because they can be degraded by proteases, and they're not stable for temperature, acid, etc. It's very easy to destroy protein function. But if you had a small molecule that could do the same thing, in general, they're much more stable. You can design them to be metabolically stable. You can design them to be orally available. So you can make them much more like a pill that you would want to take, as opposed to something that's not useful as a drug. Another possibility, again, these are all possibilities, is that you can have different selectivity. So oftentimes, let's take, for example, adrenergic receptors. So adrenaline activates, or epinephrine activates multiple different types of adrenergic receptors. And the problem is that it's difficult to modify analogs to make them selective for, let's say, one receptor as opposed to another, because they're all evolutionarily related. So the residues, the amino acid residues in the receptor that bind the epinephrine are the same. So it makes it difficult to find an antagonist, let's say, that will block one receptor but not block the other receptor, because those binding sites are conserved. However, much of the rest of the molecule may not be so highly conserved. And so you may be able to find binding sites that are present, let's say, in the alpha-1 receptor that are not present in the beta-2 adrenergic receptor. And therefore, you can design isoform selective agonists or antagonists much more easily to allosteric sites than orthosteric sites. This is not always true, but there are cases where it's true that allosteric modulators can be safer than orthosteric drugs. And one possibility is that they may be inactive in the absence of endogenous ligand. So you might have a case, for example, where what the allosteric modulator does is it amplifies the effect of the orthosteric ligand. So maybe it doesn't affect the binding, but it actually affects the downstream signaling event that's triggered by the orthosteric ligand in an amplifying way. And so that's nice, because the body is still sending out a signal, and the timing of that signal is still correct. You're just increasing or decreasing the amplitude of that signal, depending on whether it's a positive or negative allosteric modulator. Another issue has to do with desensitization. So in fact, many of the anti-androgens that are given, for example, for patients that have prostate cancer, the way they work is normally many of those hormones are released in a pulsatile manner. And so nature's designed them to be released that way so that you don't get desensitization. But if you take, for example, those hormones all the time, what they do is they actually lead to desensitization. So ironically, the hormones that are responsible for driving testosterone production, if you take them chronically, it'll actually decrease testosterone production, because unlike the normal endogenous hormones that are released in a pulsatile manner that don't result in desensitization, if you take them pharmacologically all the time, they actually lead to desensitization and turn off the very signal that their nature designed them to trigger. It's also possible to make partial allosteric modulators. So if you remember from some of your early lectures on what partial allosteric modulators are, so if you remember, a total or full agonist is an agonist that causes 100% activation of a response, and a partial agonist can only, even at a saturating concentration, can only partially activate the response. And it's not because it doesn't have high enough affinity, it's because it can't activate the downstream response from the receptor as effectively as a full agonist. So because, as we said, what we're dealing with here are changes in conformation, it stands to reason that if you can have a partial orthosteric agonist, you can also have a partial allosteric agonist as well. And so that may be useful, because maybe you want to have a partial activation or partial inactivation of a response, but you don't want to have a complete 100% effect, because that may be toxic. So it may be safer to have only a partial effect than have a full agonist or full antagonist effect. Now, of course, the use of orthosteric and allosteric drugs is not mutually exclusive, so you can combine them, and so you have much more flexibility if you combine different types of orthosteric and allosteric ligands. Now, this is more of a practical drug development point, but there are cases where orthosteric ligands have been studied for many years, and so you're unlikely to come up with a better drug than what's already out there. But for many of these receptors, there aren't allosteric ligands available. And so for the reasons that we just discussed above, there's a possibility that allosteric ligands might be better. But in this case, you're not competing with all the many orthosteric ligands that might already be out there. So if you're doing drug development, it's going to be much easier to patent something and call it your own. And of course, when you're making a new drug, it has to be better than what's already out there. So it's going to be a lot easier than if you're trying to develop just another orthosteric ligand. All right, so let's take a few examples of allosteric modulation. So in this case, we're going to talk about G protein coupled receptors, or 7 transmembrane receptors. So a lot of this comes from a very nice review article from Lindsley and colleagues, 2009. And this figure shows you a lot of what we were talking about with respect to what allosteric ligands can do, which is essentially everything. So for example, they can modulate the affinity. So in this case, you see that the orthosteric ligand binds over here, the allosteric ligand binds over here. And in the case of affinity modulation, when the orthosteric ligand binds and the allosteric ligand binds, it can actually change the affinity. So here's a binding assay down here, and you can see that with affinity modulation, so the green, so basically what you see down here is there's a shift in the concentration binding curve. So in the control, which is the orthosteric ligands alone, it has this affinity curve, and you see that with affinity modulation, it doesn't change the efficacy in this case, but it shifts the curve to the left, indicating that it's higher potency or higher apparent potency. So it actually increases the apparent potency of the orthosteric ligand. So that's the first possible effect. Another possible effect is efficacy modulation. So in this case, you can see that up here, it's affecting not the binding event, not how the orthosteric ligand binds, but actually how it leads to its response. So it's amplifying the effect of the orthosteric ligand after it's already bound. And so you can see down here that if we compare with the black curve, instead of shifting it to the left the way that affinity modulation does, it actually shifts it up. So it's actually increasing the efficacy. That's why it's called efficacy modulation. Now, there are other cases where you can have agonism by itself. So in this case, you see the allosteric ligand binds, and it by itself induces a response. And so that's what you see here with the green curve, is that even in the absence of your orthosteric agonist, you're getting a partial effect. So this partial effect here, this whatever this is, 20% or so activation, is what you're getting from your allosteric agonism. Now again, these are possible effects. So you can have, for any particular allosteric agonist, you could have combinations of these as well. But again, the important point here, so if you think about the orthosteric ligand here versus the allosteric ligand and the response, so it's not shown here, but let's remember back to what we were talking about, T conformations, R conformations. So what this is telling us is that in this case, the orthosteric ligand and allosteric ligand, they're binding different sites, but they're both causing conformational changes that's leading to a response. So again, the important point is the change in the conformation. So it doesn't matter so much if the drugs are binding at exactly the same site, but rather that they're driving the same conformational change in the receptor. All right, so this is just to show you 7 transmembrane receptor here, these helices through the plane of the membrane. And so the point is that there are many little clefts and crevices between these helices that can be either orthosteric or allosteric binding sites. And there's a lot known now about how G protein coupled receptors bind ligands, and it's known that the ligands can bind in all sorts of ways. They can bind in these crevices, they can bind in pockets between these helices outside of the membrane or within the plane of the membrane. So there are many possibilities for how G protein coupled receptors can be regulated for both orthosterically as well as allosterically. Now, this is a very important figure relating to G protein coupled receptors because it leads to the idea of what are called biased ligands. And so by biased, we mean that normally, let's say you have a G protein coupled receptor such as this one, and it binds to its particular agonist. So this would be, this little green thing here would be sort of the endogenous agonist. And normally it can activate, let's say, multiple possible pathways. So one pathway would lead to activation of adenylate cyclase and cyclic AMP production, leading to pKa activation. It can also activate the GERK kinase, leading to phosphorylation and binding of beta-arrestin, and this can desensitize the receptor, but also these complexes can trigger additional signaling pathways. So what this is saying is this one agonist can induce more than one response. So biased agonists would be designed to specifically or preferentially activate one of those downstream responses over the other. So in this case, you see the biased agonist, instead of activating both of these pathways, it only activates the beta-arrestin pathway. It doesn't activate the cyclic AMP pathway. And in this case, the beta-arrestin pathway is leading to activation of ERK. So these types of molecules have been found to be very important because, let's take, for example, heart failure. There are beta-adrenergic drugs that actually, where it's beneficial that they activate one of these pathways, but not both pathways, because if both pathways are activated, it can lead to actually worse outcome long-term. And so in this case, you want to have a biased agonist that can activate the pathway you want without activating the pathway that you don't want. So there's a lot of interest in trying to design biased agonists and trying to understand what are the different conformations of the receptor that lead to one response versus the other response. All right, so let's take a break. We've been talking for a while. Go get something to drink, walk around, do what you need to do, and then